There is a huge diversity of shape and form in the plant world. Many plants and their flowers exhibit strong symmetry, while others appear rather random. Despite first appearances, most plants conform to a relatively small number of simple, repeating geometrical patterns. Spirals are a common pattern seen in many plants and their parts. You'll be familiar with the spiral pattern of the pineapple and in fir cones. Here there are two different sets of spirals, right-handed and left-handed. We'll concentrate on leaves and flowers to determine the nature of these patterns and see what research can tell us about the development of such patterns in plants. To determine the patterns in which leaves are produced, we need to look very carefully and to be aware that all may not be as it seems. This palm appears to be composed of pairs of leaves, regularly arranged in a plane, slightly curved by the effect of gravity. But these are not pairs of leaves, they're sections of a single leaf. This becomes much more evident when we look at other, less completely serrated leaves. To see the pattern in which leaves are formed, it's often easier to look at the pattern formed by the stumps of old leaves. Each branch of this tree fern is a single leaf, and the leaf scars on the trunk show a spiral pattern. An essential feature of the spiral is that the leaves form singly and do not exactly overlap each other. Here, the increasing size of the leaves, starting with the youngest in the center, picks out the sequence of the spiral and the angle between subsequent leaves, which is usually about 137 degrees. In addition to spirals, there are two more common forms of leaf phyllotaxis. These leaves lie in a plane, so they're not spiral. They form a zigzag pattern known as disticus. In disticus phyllotaxis, leaves form singly, and the next leaf is offset from the last by an angle of 180 degrees. Here the leaves are formed in pairs. This is whorled phyllotaxis. The commonest form of whorled phyllotaxis is decussate, where leaves form in pairs which are offset from the next pair by an angle of 90 degrees. A rarer form of whorled phyllotaxis is tricussate, where sets of 3 leaves are formed, offset from their neighbours by an angle of 60 degrees. These 3 types of vegetative phyllotaxis, spiral, disticus and the whorled forms, cover virtually all the leaf patterns of plants, and the great majority of plants have spiral phyllotaxis. Flowers present a bewilderingly huge and marvellous variety of geometrical forms. Despite the apparent diversity, most flowers can be classified into just 2 types of geometrical pattern, spiral and whorled. The key to sorting out which is to appreciate that most flowers have become highly modified structurally to create efficient mechanisms to lure insects and achieve pollination. Most of these modifications involve the fusion and distortion of petals to form platforms, tubes, even sprung gates. And of course, plant breeders have artificially selected a large variety of flowers with the express purpose of accentuating some of these features. So, although the petals may form apparently distinct geometrical types, these are not the patterns in which the various organs of the flower develop. As with leaves, so with flowers, all is not always what it seems. Multi-headed flowers are quite common, but some are not so obvious. This is not a single flower, rather a composite flowering head. The yellow centre is composed of a large number of small bisexual flowers. Each apparent petal is in fact a single female flower made up of 3 fused petals which has been adapted to act as a lure for insects. Such composite flowers form a very large family which dates back to Miocene times, but this is comparatively recent in the long history of the flowering plant, and these are considered to be advanced flowers. At the other extreme is the oldest and most primitive group of the flowering plants, the subclass Magnoliidae. The magnolias are modern members of this primitive group. They have few simple petals and a large number of male and female internal organs, stamens and carpels. A similarly ancient family is the Ranunculaceae, familiar today as buttercups and anemones. They too have simple petals and a large number of internal organs. This ensures a high probability of pollination by insects wandering over the flower. By contrast, many of the more advanced flowers have distorted and fused petals forming a lure and a pollinating mechanism finely attuned to an insect approaching on a very precise path. Thus, the large number of internal organs gives way to a small number of precisely placed stamen and often united carpels in the form of a pistil. Another feature of the advanced flowers is the regularity of the number of organs that make up their flowers. All the flowers of this succulent are identical. They have 5 green bracts, a bell of 5 petals, 10 stamen and 5 carpels at the centre. The flowers of other members of this family all have a similar small and regular number of organs. In contrast, primitive flowers have less narrowly defined organ numbers with variations occurring even in flowers on the same stem, and organs such as stamen varying in number from less than 10 to well over 1,000 in flowers of different members of the same family. This variation can clearly be seen in the number of carpels, the fat organs at the centre of peonies. It's very difficult to determine the phyllotaxis simply by looking at the flower in full bloom. The development of the phyllotaxis is seen more clearly in the flowering meristems, but these are minute. First, the enclosing leaves must be stripped away. Then the developing organs, the flowering primordia, are revealed. The family Asclepiadaceae contains some of the most highly modified advanced flowers. In Ceropegia, a member of the same family, the petals are fused and contorted into an elaborate lure for pollinating insects. The Swiss botanist Peter Endres has studied the floral primordia of a very similar member of the family, Asclepias curasavica, and finds that development of the meristem leads to extraordinary synorganization between neighbouring stamens and carpels. Clearly, studying the development of the floral primordia reveals a lot about the development of the phyllotaxis and the fusions that form the complex flower. But is the phyllotaxis in any way connected with the evolution of the flowers? Does it distinguish between advanced and primitive? The buttercup is a member of the primitive flower family Ranunculaceae. This is its flowering tip, the floral primordium. Each of these lobes or primordia will develop into an internal organ. Carpels on the inside and stamen outside. By careful study, Endres has been able to determine the sequence in which the primordia formed. Here they're numbered in reverse order. 1 is the youngest primordium. This is the developmental sequence, but does it have a pattern? There are several, but they are all spirals, reflecting that the underlying developmental sequence is itself a spiral. Aquilegia is a member of the same primitive family, but has a more symmetric and modified flower. There are 5 organ primordia at the centre, then 10 files of overlapping primordia. Looking more closely, we can see that these are in fact alternating sets of 5 primordia. This is whorled phyllotaxis. So the 2 principal forms of flower phyllotaxis, spiral and whorled, are both seen in primitive flowers. Nevertheless, whorled phyllotaxis is dominant in advanced flowers. Endress believes that there are 2 reasons for this. Firstly, the symmetry of the whorled form is a prerequisite for organ fusion. And secondly, spiral phyllotaxis, which is an advantage in arranging large and variable organ numbers in primitive flowers, is a distinct disadvantage to obtaining optimal spacing with the low and fixed organ numbers of advanced flowers. But whatever the evolutionary significance of plant phyllotaxis, a fascinating question remains. Why do plants conform to such a small number of dominant phyllotaxical forms? Paul Green of Stanford University.
The overwhelming majority of vegetative phyllotaxic patterns fall into 3 categories. Spiral, as seen in this ficus. Disticus, or zigzag in a plane, visible in this clivia. It's also present in ivy, although this is less obvious because the leaves are twisted so as to catch more light. The 3rd category is whorled, as illustrated in this decussate plant, where the leaves are present in pairs and opposite, and these pairs are at 90 degrees to each other. Each type of plant must have a developmental engine to produce the pattern. This must be located at the site where the new organs are produced, which is at the tip of the plant. Our search, however, is not for 3 different types of machine, but rather a single type which can operate with variations. The reason is seen in this bromeliad, where in the basal vegetative part, the arrangement is clearly spiral. The inflorescence, however, is clearly in the zigzag or disticus arrangement. This same capacity to shift from one pattern to another is also evident in vegetative parts of the plant. This eucalyptus has its basal leaves in pairs. Toward the tip of the shoot, however, the leaves are clearly arranged in the disticus or zigzag in a plane pattern. Although such changes in phyllotaxic pattern are not all that common, they do illustrate that the same developmental engine can switch from one pattern to another. The same capacity to generate different patterns from the same basic device is illustrated in the spirograph. Here, the disticus form, zigzag in a plane. Here, decussate, one of the whorled forms. And finally, the spiral form. All of these patterns were made with the same basic apparatus. The only variation was in the small inner rings and the number of cogs on each. If there is a basic mechanism to generate all patterns, it has to be found at the tip of the shoot. I've made an extensive study of this region. To do so, one has to expose the apical dome by careful dissection, and then remove the surface of that dome in order to examine it in the compound microscope. The cells away from the center are all aligned parallel to each other in files. If we look at this under polarized light, something else is revealed. The cells darken and lighten again as the specimen is rotated because the cells contain parallel bands of cellulose microfibrils. The cells are cylindrical, and the cellulose microfibrils run along the circumferential direction, much like hoops on a barrel. In polarized light, when the cell is in one orientation, this has as a consequence that the transmission of light is prevented and the cells appear dark. At 90 degrees to that, the opposite occurs and the cells appear bright. Looking at the entire layer taken from the apical dome, you can see that the fibrils are oriented in two major directions at right angles. Change the polarization and the pattern reverses. The significance of the cellulose microfibril alignment is that it imparts a strong directional stiffness to the cells and tissues. That can be illustrated in a metal watch band of this type, where the cells correspond to these vertical lines and the cellulose reinforcement is at right angles. This band is quite flexible in one direction and very stiff at right angles to that. This band as a whole could be called a reinforcement field. To study the details of the cell alignment, I use scanning electron microscopy. Rather than use a fixed or dehydrated meristem, we actually make a mold of the living apical dome itself. The cast that emerges is a faithful reproduction of the meristem. This can then be gold-coated and observed in the scanning electron microscope. This is a scanning electron micrograph of a decussate apex. These two areas are stumps of the youngest leaves. The remaining area is the formative apical meristem. You'll notice that in the center the cells are relatively disorganized and would not constitute a coherent reinforcement field. On either side you see straight cell files, which would represent reinforcement fields. The question is, how could these fields lead to the production of new leaves? The next ones to appear are expected to arise here and here. The watchband analog can shed light on this. The class represents the area in the center of the apical dome where there's no clear cell pattern or preferred direction of stiffness. The band itself represents a reinforcement field. Because the cells tend to grow, stresses develop in the reinforcement field, and a buckling ensues, which produces the new primordium. Here is the same decussate apex at a later stage. You notice that the reinforcement field has buckled and produced a primordium on this side as well as on the other. It's also evident that new reinforcement fields are arising at right angles to the previous one. They will form the next pair of primordia. So in general, recognize that the apical dome is a composite of several reinforcement fields, and it's their behavior that determines the phyllotaxis. Turning now to the disticus apex, this one is of ivy, we see a related situation. There are, however, several reinforcement fields involved, and the new leaf is starting to appear as a kind of mechanical compromise between the three fields, the resultant putting the leaf on the same axis as the previous leaf, which is shown at the bottom. Here is the disticus apex at a later stage, and you see that the previous small ridge has developed into a quite easily visible primordium. The next leaf to come has its precursors here in a reinforcement field that you can barely see, almost obscured by this older leaf. The resultant is that the leaves form the disticus phyllotaxis. Here we have a spiral apical meristem. The oldest leaf is here, with its tip removed to allow visibility of the dome proper. The next younger leaf is here, younger still is here, and a primordium just emerging is seen here. These leaves are offset at the Fibonacci angle, which is 137.5 degrees, running from this leaf to the next younger leaf. You'll notice that each leaf has a reinforcement field running outward from its center. And as before, the apical dome proper is a composite of a variety of reinforcement fields. The theory is that the next primordium to emerge does so because that location is a resultant of all the forces and all the directional structures on the dome.
The generative motor in the plant meristem seems to be a mechanical strain field in which cell growth and cell division occur in such a way as to minimize the strain fields. This is the process that Paul Green has suggested to give rise in a natural way to the basic plant forms, decussate, disticus, and spiral. Now, if that's correct, then it should be possible to produce a computer model that will in fact generate these forms and no others. Now, the plant meristem is a very complex system to model, and my colleagues and I, that is Christian Briere and Lynn Traynor, have been exploring these ideas on a totally different organism, the unicellular green alga, Acetabularia acetabulum. Acetabularia grows into a characteristic adult shape through a series of forms by a process that has basic similarities to the growth of the higher plant meristem, despite its being a single cell. The tip, of the order of 1/10 of a millimeter in diameter, grows about 1 millimeter per day. Every few days, this tip, normally conical in shape, flattens, and a ring of small bumps is produced. These grow into delicately branching structures called bracts, or verticils. They are like little pointed leaves. The whole ring of verticils is called a whorl. After several of these whorls have been produced, a slightly different shape emerges, a cap primordium. This then develops into the mature cap with its parasol-like structure. The whorls then drop off, so they don't have any function in the adult. This gives us the adult form of the alga, which lives for many months before reproducing. The structure of a whorl is one of the basic leaf patterns in which leaves are produced periodically as the plant grows. In our model, what we've done is to describe the mechanical properties of the cytoskeleton, that is the network of filaments in the cytoplasm, in interaction with calcium, calcium being one of the major ions concerned with plant morphogenesis. These then interact with the cell wall to generate changes of form. So what we've got is a general purpose model here, not specialized for any particular organism. The system starts with cell variables at constant values throughout the dome, so that there is initially no spatial pattern at all. The dynamics of the cytoskeleton-calcium interactions then spontaneously generate a pattern, with calcium, indicated low by green and high by red, rising to a maximum at the pole. This initiates the growth process. As the tip extends, the initial peak of calcium at the pole changes into a ring of elevated calcium, seen as a red annulus around the tip. This annulus increases and decreases in a somewhat irregular manner, also changing its position as growth proceeds. If this annulus were to break up spontaneously into a ring of peaks in calcium concentration, then we would have produced evidence in favor of the model, supporting Paul Green's hypothesis. Therefore, we tested it. Precisely this result occurs. Whenever the annulus reaches a sufficiently high concentration, it breaks up into a series of peaks. The whole sequence of events in the model is associated with changes in the pattern of calcium. The model is effectively predicting this correspondence between calcium patterns and morphogenesis. Finally, as growth proceeds, the model generates a new form. Instead of a well-defined annulus of calcium, the concentration becomes elevated over the whole tip, which then grows into a bulbous form, not exactly a cap, but a terminal pattern. Experimental observations have shown that the calcium concentration is very similar to the predictions made by the model. But one of the most important results of the model simulations is the demonstration that one of the basic leaf patterns, the whorl structure, is generated perfectly naturally, almost without trying. It is a natural consequence of the dynamics of the model itself. And so whorls are produced even though they serve no function in the adult organism.